Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is a place where we make the learning of physiology easy, exciting, and effective. Thank you for joining me. And if you're new to this channel, you're especially welcome. And if you enjoy and like the content that we share, kindly click the like button. Also click the subscribe button. And don't forget to turn on notifications so that you don't get to miss any new content that we drop. All right, let's get right into it. So today we'll be dealing with the part 3 of digestion and absorption. And it has to do with the digestion and absorption of lipids, or you can call it fats. All right, so the most abundant lipids that we ingest, that's dietary lipids, they usually they are neutral, we call them neutral fats. Neutral fats. And they come mostly in the form of triglycerides. Triglycerides. Okay? So we can also have others like cholesterol. Cholesterol, we also take some cholesterol. Phospholipids too. Phospholipid, but this is the most abundant dietary fat. So we'll talk a little more on this. So what happens is that the digestion first of lipids is not as straightforward as that of carbohydrate and proteins. Then carbohydrates just have the amylase that breaks it down. Then you have the disaccharidases that also break it down further to release the monosaccharide. So it's straightforward. Okay, you have the proteases and dipeptidases and all of that. They do their work in a very straightforward manner. But now, in the case of fat, the lipases alone do not do an effective work. I remember we mentioned a little of that when we were talking about the secretions, gastrointestinal secretions. And this is how it is. Fats, they usually coalesce, they usually join themselves together into blobs or lumps. Okay? A fatty lump like that. So what happens? The main challenge, why the digestion of and absorption of these lipids is unique, is the fact that it's insoluble in water. You know, a lot of water, electrolyte, is secreted into the intestine. And lipids or fats and water do not mix together. Do you understand? So the watery environment in the intestinal lumen poses a problem. It cannot now go easily to touch the villi. Okay? Villi is here like this. It doesn't find it easy. Everywhere is covered with water. So it cannot easily go to the intestinal villi for absorption to take place. So both the digestion and absorption is a challenge. So how does the body solve that problem? It is called bile salts. That is the solution. And the reason why bile salts are so effective. You know, bile salts, we said that one of the very major action it does that aids digestion. They do not digest lipid, but they help the digestion of lipid in a very physical way, which we said is emulsification. Emulsification. And let's really understand what this emulsification is all about. We also call it detergent action. You know, it works like soap. You know, if you put your hand, carry, let's say, groundnut oil, stain your hands with groundnut oil, and you go under a tap to wash it, will it go? No. You use just without anything, water, and to try to wash the groundnut oil off your hand, it will not go. But when you put soap, it just washes everything up. How does that soap do the magic? It's the process of this emulsification. Okay? Because soap in itself, there's what they call, in those days in biochemistry, it was saponification. Saponification, that's the process of soap making. So the process of soap making is by mixing fat plus a base or an alkali. Okay? It now leads to soap. So this process is called saponification. So soap is a mixture of fat and alkali. So soap in itself has a fatty part and also the water-soluble part. So it has hydrophobic part, hydrophilic part. So the main thing that makes soap easy is that it can relate both with water and can relate both with fat. So it uses the fatty aspect to relate to that oil and through physical shaking. Okay, that's why you need to wash your hand. It's called agitation. Through physical force, it removes it from the body and also introduces water to wash it off. So that's just a very easy way to understand. So the same thing, bile salts are what? They are amphipathic. That is what gives them that ability, just like soap. They are amphipathic. Okay, so they have the hydrophilic part, which is usually represented by head, then hydrophobic tail. This is the hydrophilic head, then hydrophobic tail. So this amphipathic nature of bile salts, it's what makes them able to do this to the fats. First of all, they help to break down. We say this is the fat in this big lump. If the lipase comes, the lipase can only touch the surface. It cannot touch the rest of it. So a lot of it will not be digested. But when bile salts are introduced through the amphipathic activity, they can break into this thing. And because they have both water and hydrophilic and hydrophobic parts, the hydrophobic part will break into it, and it just has a way of breaking it into smaller pieces. Okay? And it breaks it. So by so doing, it increases the surface area. So a lot of the fat the lipase can now touch more surface. That's what it means by increasing the surface area. So the only part that will not be touched is this small part, but it has touched a lot. So it's just a little of it that will not be digested. Okay? So that is what happens. So it has helped in the digestion now. And the digestion is what? This is the most abundant. So it breaks triglycerides into glycerol. Breaks it and you get glycerol, monoglyceride, and you can also get fatty acids. So these are the three major products of lipid digestion, of fat digestion. Okay, we break down products as a result of lipase activity, especially pancreatic lipase, because it's from the pancreas you start having the bile salt. The pancreatic duct connects with the common bile duct, okay, to get the pancreatohepatic duct. So that is from the pancreas. So lipid digestion, the lingual lipase that is produced in the mouth, gastric lipase, those ones have very little effect because of the fact that they cannot be broken down. The surface area is too big, so it's negligible. The main digestion of fat happens in the small intestine. So now, the bile salts have helped through emulsification to digest it. So So what does it do? How does it now help in the absorption? Because absorption is also a challenge. It cannot easily cross and enter the enterocyte to be transported into the blood. So we are going to learn the mechanism of the absorption of lipids after this break. All right, welcome back. So now we're going to be dealing with the absorption of fats. Now, this is just a simple schematic diagram to represent a micelle. Okay, this is a micelle, it's called a micelle. And what is this? This core here, this is the lipid core. Okay, so the micelles, the tail, the hydrophobic tail. Okay, hydrophobic tail. Then this is hydrophilic head. Okay, so now this is how the micelles, they organize themselves. They surround. Now it has broken it down. It has broken the triglycerides. It has been broken down to these simple forms, fatty acid and monoglycerides. So now, because the monoglycerides itself, they are still lipids now, just that they are now smaller, okay, in the smaller form to pass through, to be absorbed. But then it is still not soluble in the watery environment. So this the water-loving part faces outside. So it covers it. So it's like a cheat. You understand? It's like a cheat. It forms a new entity that makes it look as if it is water, as if it can dissolve in water. So this hydrophobic fat dissolves and mixes well with the water. So what it does is that the enterocyte like this, these are the cells, enterocytes, it can now easily get to the membrane of this entire. When they get to the membrane, it has brought it. So it does what is called like a ferrying, call it ferrying action. That means it's moving it just like a boat. It carries it close to this point. Then when it gets very close here, then it now stays back. It now puts it into the enterocytes at this place, and it goes back. You understand? So the micelles, all these individual bile salts, they don't follow it inside the enterocyte. They only bring it close, because when this whole place is covered with water, and this thing cannot, because it's insoluble in water, it cannot get close enough. It repels, hydrophobic. The water repels it. It just cannot get close. So what this one does is to carry it to get to the tip of this enterocyte, and then it pushes it into the enterocyte, and itself does not enter. Remember that at the jejunum, where this thing really happens, the absorption of fat happens, the micelles do not go into the enterocyte. But when enterohepatic circulation, remember we'll talk about enterohepatic circulation. So enterohepatic circulation is talking about the fact that these bile salts will later be absorbed. So where bile salts are absorbed is at the ileum. Very important point to note. So when they have finished doing their job in this duodenum and jejunum especially, they've taken all the fat absorption happens in the jejunum. Then they don't have anything to form micelles on. Then they themselves in the ileum, they now get absorbed and enter the portal circulation, which goes to the liver and resecretes it again. So this is just what happens. So now this breakdown product of fat, they've entered the enterocyte. But then something happens. They now get what? Re-esterified. Re-esterification happens. Re-esterification of the fats. So re-esterification of fat, what does it mean? It simply means that these breakdown products that have been broken down, what happens? They now recombine to form these triglycerides. It's a very funny thing, fats. Very funny. So the breakdown is just to allow it to be small enough, because when it's in a lump form, it's too big. There's nothing, even the micelles, it cannot just cross. Okay? So it's just to make it small enough to cross into the enterocytes. But the moment they enter into the enterocyte itself, right in the enterocyte, they recombine to form this. They now go back to their pre-digested form. You see it? The form of triglycerides again. So they are now in that big form of triglycerides. What now happens? The Golgi, they are now packaged in proteins to form what is known as chylomicrons. From the Golgi apparatus, they form chylomicrons. Okay? They are packaged in chylomicrons, surrounded by proteins. So after they have become packaged as chylomicrons, what happens next? Now, because they have become so big in big chylomicrons, of course, they cannot cross the basolateral membrane. Remember, we have the apical membrane. So the basolateral membrane is where they cross to enter the blood. But they cannot cross. They are too big. So the way they can get out is through exocytosis. Remember, exocytosis is one of the ways transport across cell membranes. But it's when you have macromolecules, very big molecules, they use exocytosis. So it uses exocytosis to cross and now enter the interstitial fluid. But it now needs to cross to enter the capillaries, to enter the blood. But that is another what? Problem. It's too big to even cross the capillary membrane. But there's something else. They are called lymphatic capillaries. Okay? It's what we call lymphatic capillaries. They are not the same with normal capillaries. Normal capillaries are from arteries. Arteries form capillaries, and capillaries later come together to form venules and veins. But these lymphatic are totally different. And they are called lacteals. These lymphatic capillaries, they are called lacteals. So these lacteals, they have enough space like this for it to pass. It can pass through. They have pores, holes, that these chylomicrons can easily pass through. It cannot pass through normal capillary. So the process of absorption into the circulation is through lacteals, chylomicrons. All right? So when they enter the lacteals, the lymphatic circulation, you know, the lymphatic circulation, in the end, they all empty in the thoracic duct. Okay? So they empty in the thoracic duct, which now later empties into the inferior vena cava, and it now enters the real blood circulation. So that is how it now enters the normal circulation, and fat absorption is now said to be complete, enters the bloodstream and distributed to wherever they are needed. Okay? So this is the digestion and the absorption of lipids. It's a little more technical than the rest. Okay? But this process of entering, being packaged chylomicrons and entering lacteals and thoracic duct is usually for the fatty acids that are longer than 12 chain, larger than 12 chains. 12 chains of fatty acid. Very important to note. Okay, so the smaller chain fatty acids, they can easily cross the capillary. But most of these large chain fats, okay, this is the route most of them follow, long chain, because they have been re-esterified, they now become longer. But some are still short enough to, and small enough to pass through the normal. So you see, that's how it is. It's not so difficult after all. All right? So I've written a book on gastrointestinal physiology, said broken down for you. Very easy for further reading. And also written some other books in physiology. 7 of them as at the time of this shooting. So the links are all there in the description box. You can click it to download any one of your choice to read. For further reading, go through more details of these things are found there. Okay? If you also check, you see the link to our website, so you can get informed of other things we are doing to make the learning of physiology easy, exciting, and effective. See you in the next video.